All right, we're going to go through some more biochem things. So these are on Moodle, all these PowerPoints, but they are from a different text. So it's going to say chapter 14. It is not chapter 14. All right, just a few more things about carbohydrates. Here, you got carbohydrates, major source of energy from our diet. It's called carbohydrates because it's carbon with some number of waters. So this is whatever, X and Y. If you have glucose, you have C6H12O6, or it is C6 with 6 waters. So you have a carbohydrate. All right, also called saccharides, which means sugar. They're produced by photosynthesis in plants. You got glucose from CO2 and water, okay, and the energy from the sun. All right, so you got your carbohydrates, and then you have animals, and then you have a respiration, and you have the nice cyclic process there. All right, so they are oxidized in living cells to produce CO2 and water, and then it just keeps going and going, except at this point we're producing more CO2 than can be used for photosynthesis. All right, so types of carbohydrates. You got your monosaccharides. All right, so that's the monomer of your polysaccharides. And if you take two monosaccharides, then you have a disaccharide. So monosaccharides are the simplest of your carbohydrates. And then, of course, your disaccharides, you're going to have two monosaccharides, and a polysaccharide, many monosaccharides. All right, so if you have your disaccharide, you can hydrolyze it. Hydrolysis means split it with water, usually in an acid catalyst here. And so if you take the most common disaccharide, which is sucrose, and you hydrolyze it, you get a glucose and a fructose. All right, so if you got your polysaccharides, you can hydrolyze that with many waters to get many monosaccharide units. But monosaccharides cannot be hydrolyzed. They are the simplest carbohydrates. All right, so your monosaccharides consist of 3 to 6 carbons. Those are typical monosaccharides. And they have a carbonyl group, either an aldehyde or a ketone. And so yesterday we saw that glucose is an aldose. It has an aldehyde group. Fructose is a ketose. It has a ketone group. And we saw on both of them several OH groups. So the hydroxyl groups are your OH groups. All right, so again, you will need to know the structure of glucose and fructose. All right, so again, if you have an aldehyde group, then you're called an aldose. That is a sugar ose that has the aldehyde functional group. And if you have 3 carbons, you're a triose. Right? 4 carbons, tetrose, 5 pentose, 6 hexose. And so yesterday we saw that glucose and fructose, those were our hexoses. And we also saw that ribose was a pentose. All right, so if you have an aldose, again, you're going to have the aldehyde group. And so the one listed here is 1, 2, 3, 4. This is an aldotetrose because it has an aldehyde group and 4 carbons. All right, so this is the one that we had from yesterday. This is our fructose. Except that is not fructose, okay? All right, so this is a ketohexose, but this is not the structure for fructose. All right, so you have your ketone group, you have many OH groups. Again, here you have a ketone functional group, and that is a ketose. All right, and so we're just going to distinguish between an aldose, ketose, and count how many carbons. So, okay, you got your aldehyde, so you have an aldose. And this is an aldo with 1, 2, 3, 4, 5, 6. That's an aldohexose. And here is a ketone, and this is 1, 2, 3, 4, 5. This is a keto, pentose. All right, so ketone group, 5 carbons, keto, pentose. All right, and for whatever reason, these are all separated. All right, so your structures of your monosaccharides. All right, so we are going to represent our sugars with a Fisher projection. All right, and so a Fisher projection is just a plus, right? And so, and it is understood that these on the horizontal sides are coming out, and the ones that are vertical are going back. So this is back, and this is out. All right, so when you're looking at a Fisher projection, you're looking at a chiral carbon. All right, and so anytime you're doing your carbohydrates, the aldehyde will always be on top, or your ketone is going to be very close to the top, as close to the top as possible. And so you're looking at your bottom chiral carbon here to determine whether it is a D class of sugar or an L class of sugar. If your OH is on the left, then it is an L class of sugar. And then, of course, if you take the mirror image, then you have the OH on the right side, which is your D class of sugar, and the naturally occurring sugars are the D class of sugars. All right, and so you only put the plus. This cross indicates that this is going to be a chiral carbon. So this carbon here is a chiral carbon, which means it is a carbon connected to 4 different groups. All right, so you want the chiral carbon furthest away from the carbonyl group. So the one the furthest away from the carbonyl group, the one with the highest number, furthest away. Again, if it is on the left, it has the capital L. If the OH is on the right, it has the capital D. All right. All right, so they highlight it. Here's your final chiral carbon, the one furthest away from the C double bond O. All right, OH is on the right. That is your D glucose. All right, and so here your OH is on your right. Here is your D ribose. Both of those you need to memorize the structures for. You do not need to know the galactose, but here is the galactose. It is the L galactose because your OH this time is on the left. All right, so that's just how you distinguish between a D and an L. And the D are the naturally occurring sugars. All right, so very simple. OH on the bottom chiral carbons on the left. OH on the bottom chiral carbons on the left. OH on the bottom chiral carbon is on the right. And so there are the answers to those. Here is the correct formula for D-fructose. And if you look back at the other one, you'll see that on the other one, they had OHs all on this side. That is not D-fructose. This is D-fructose. You got your OH for a D has to be here, and then another one, and then the third chiral carbon on the way up, the OH is on the left. And that is the structure of D-fructose. All right, D-glucose. All right, so again, the glucose. So what you have to know is D-glucose is an aldose. You have to have the aldehyde group on top. And then you need to know the order where you put the OHs. So you got 3 of them on the right, and you have to know it is the first one, the third one, and the fourth one. And the second one is on the left. And all of them end in a CH2OH. All right, and so that's how you would memorize your D-glucose. All right, also known as blood sugar. It's also known as dextrose. Let's say that anywhere. Okay. It's the monosaccharide in polymers of starch, cellulose, and glycogen. Right, so you always have your test, your blood test. They'll test the glucose levels in your blood. Okay. Hyper, too much. Normal, hypo, not enough. All right, so you will have your blood test, but you should know that hyper is high, hypo is low, and of course, then you would have your normal group. Fructose, again, you'll notice that the fructose got rid of the chiral carbon here from the D-glucose. That is now a C double bond O. But the OHs are the same. On the third carbon down, it's on the left, and then it's on the right. So these do not change. These are the bottom 4 carbons are exactly the same as they are for D-glucose. It is the top that has changed. All right, so it's ketohexose, sweetest carbohydrate found in fruit juices, converts to glucose in the body. So there is a mechanism when you do bioorganic, you'll have the mechanism for converting glucose into fructose. All right, galactose. All right, so when we had our OH and our OH and our OH and our OH. All right, so that's where we have our D-glucose. So you have that memorized. You do not have to memorize D-galactose. However, if I want you to give me the structure of D-galactose, I will tell you how it's different than D-glucose. It is not different here. It is not different here. It is different right here. It is not different here. All right, so if I look at my D-glucose, my glucose has the OH on the right, and then the OH on the left, and then the OH on the right, and then on the right. So the only one that is different is this carbon, which is carbon number 1, 2, 3, and 4. So it is different at carbon number 4. All right, so that's what it says right here, similar structure to glucose, except for carbon 4. And so this is called a C4 epimer of D-glucose. So if I say D-galactose, give the structure, I can't just say that. I have to say D-galactose. It is the C4 epimer of D-glucose. All you got to do is write out D-glucose, and then on the 4th carbon, change your H and your OH. That's what epimer means. All right? It means that's where you're going to switch your H and your OH. C4 tells you the number of that carbon to do it. And that is D-galactose. So it'll say D-galactose, the C4 epimer of D-glucose. And so again, you write your D-glucose that you have memorized, and then you change the H and the OH at C4, and now you have D-galactose. All right, so it is obtained from lactose, which is a disaccharide. So when you are lactose intolerant, all right, what you're intolerant of is the D-galactose in your lactose. And you don't have the enzyme to break down your D-galactose. So your lactose was broken down into D-galactose and D-glucose. All right, so here's your D-fructose. Again, you will see that the bottom, this is the exact same as your D-glucose. All right, next. Gotta find a way to put these together. All right, so when we did our RNA and DNA, we saw that the ribose in the RNA and DNA was not in a straight line. It was in a cyclic conformation. And so we're going to see where our cyclic structures are for our D-glucose and our D-fructose. All right, so if we have our D-glucose, and so again, OH here, here and here. All right, so when we do our D-glucose, you're going to line it down like this. Your C5 hydroxy is going to attack your C1 carbonyl, and you're going to have your cyclic structure. All right, and so if you are asked to draw D-glucose, you'll draw it in a straight chain. It may say draw the cyclic form of D-glucose. And so it is a six-membered ring. All right, so here is carbon number 1. Carbon number 1 used to have the double bond O. All right, now it does not. Now it has a bond to oxygen and another bond to oxygen. That is called the anomeric carbon. This is a carbon that is bonded to two O's. There's no other carbon bonded to two O's. Carbon 2 is bonded to one oxygen. Carbon 3 is bonded to one oxygen. Carbon 4 is bonded to one oxygen. Carbon 5 is bonded to one oxygen. Carbon 6 is bonded to one oxygen. Only carbon 1 is bonded to two oxygens. All right, and so you're going to have your choice for your OH here. So you're either going to have your OH down or you're going to have your OH up. And so if it is OH down, it is called, so this is called the alpha conformation for your anomeric carbon. So you're going to have an alpha D-glucose and you're going to have a beta D-glucose. If your OH is up here, all right, so it could be down, that's alpha. It could be up, and that's beta. When they wrote is down. Everything else is exactly as written. All right, the C2 OH is down, C3 OH up, C4 OH down, C5 the CH2OH is up. That always is the same for D-glucose. All right, but your OH on carbon number 1, the carbon connected to two oxygens. If it's down, it is the alpha form. If it is up, it is the beta form. All right, okay, so they're showing that here. Again, if it is down, you have the alpha D-glucose. If it is up, you have the beta D-glucose. Everything else is exactly the same. CH2OH, yes, yes, OH down, OH up, OH down. All the rest are the same. If they are not the same, then you're not doing glucose and glucose. The only difference between this glucose and this glucose is where the OH is on your anomeric carbon, the carbon connected to two oxygens. All right, and so if you have, again, it is the C5 hydroxy that I showed on the other picture. That is attacking this carbonyl, and the electrons are going to the O. And so then the H here is now connected to this O right here. So you see that this H is now right here. All right, and so you have your chiral carbon here. And so the chiral carbon can either have the OH that is slanted downward or the OH is slanted upward. They are different because that is a chiral carbon. All right, but again, off of carbon 2, it's down and then up and then down, and then your CH2OH is up every time for D-glucose. All right, and so when you do that, when your C5 attacks here and your H from here goes to here, this can arrange where the OH is down, and that's alpha, or the OH is up, and that's beta. Now, beta is more stable than alpha, and so you're going to get more beta, 64%, than you will alpha at 36%. But you get a significant amount of both alpha and beta D-glucose. All right, fructose is going to work very similarly. All right, except now we have a 5-membered ring. All right, and so now you're looking at this carbon here, and again, this is now carbon number 2. So this is carbon 1, this is carbon 2. This is the carbon we're looking at because we're looking at the carbon, the only carbon, that is connected to not just one oxygen, but two oxygens. It is the only carbon that is bonded to two oxygens. It's got one here, it's got one there. All right, so that is the anomeric carbon. And so if your OH on that anomeric carbon, which means the carbon connected to two O's, If it's going down, then it's alpha. If it's going up, and then it's beta. All right, if you want to draw the cyclic form of alpha D-galactose, remember that is the C4 epimer of D-glucose. So we had the OH that was down, we had the OH that was up, we had the OH that was down, we have the CH2OH that is up, and then we have our O, and then we have our anomeric carbon. All right, and so they want alpha, and so we know that on the anomeric carbon, the OH is going to go down. That makes it alpha. But this is alpha D-glucose. They're asking for the alpha D-galactose. On the test, it's going to say that is the C4 epimer of D-glucose. So when you count over 4 carbons, 1, 2, 3, and 4, what you have to do is you just change that. Now this OH is not going down, this OH is going up. All right, and so you just change the direction of the OH on the designated carbon. Everything else is the same. It is alpha. This one has to be going down. And so when you look at your C4, all right, C2 down, C3 OH up. And now instead of down at C4, it is going to be up because it's asking for the D-galactose. So when you see your C4, it is going up. And of course, this tells you it's alpha, OH going down. Every single glucose is then on C2 down, C3 up, and then down. But this is galactose. That's why we had to change the C4. CH2OH up. So if you know glucose, then you will know all of them because all of them will be named as an epimer, which means change the stereochemistry at that position. All right, chemical properties of monosaccharides. All right, so reducing sugars. All right, so reducing sugars are monosaccharides that oxidize to give a carboxylic acid. All right, so we want the sugars that are reducing agents. All right, so we want to reduce something, so we got to find a reducing agent. We want to reduce something, we got to find a reducing sugar. All right, and so again, the reducing agent is oxidized. The reducing sugar is oxidized. All right, and so it's going to oxidize to a carboxylic acid. And so you have your C double bond OH, that is your aldose, and you're going to oxidize that into a carboxylic acid. All right, so if you oxidize your monosaccharide, that means this is an oxidation. All right, and so this is your oxidation half reaction. That means the reactant is called a reducing agent. And so any of your aldehyde groups are going to be reducing sugars. All right, and so to test that, you do a Benedict's test to see if it is a reducing sugar. All right, and so they include the monosaccharides, glucose, galactose, and fructose for your reducing sugars. And so again, you've got your D-glucose, which is an aldose, your aldehyde. Again, the definition of oxidation for organic and biochem is going to be gain of oxygen or loss of hydrogen. And this, of course, this carbon was bonded to 1 oxygen. Now it's bonded to 2 oxygens. This is a gain of oxygen for the oxidation. All right, and so we have oxidized our aldehyde to our carboxylic acid. And so this is a reducing sugar. Why is it called a reducing sugar? Well, we started with Benedict's solution, which is a copper plus 2. And over here, we have a copper that is a plus 1 oxidation. So this sugar reduced copper from a plus 2 oxidation to a plus 1 oxidation. And so it is therefore called a reducing sugar. Because it reduced the copper plus 2 into a copper plus 1. So just like oxidation reduction, it's an oxidizing agent, yes. But since it's a sugar, we don't call it an agent anymore. We call it a sugar. It's a reducing sugar or an oxidizing sugar. This one is a reducing sugar because it reduced copper plus 2 to copper plus 1. It's a sugar that reduced it. All right. You can have reduction of your monosaccharides. So we can oxidize them. We can also reduce them. All right. And when we take our aldehyde and we reduce our aldehyde, we get a primary alcohol. So when it is reduced, you get a primary alcohol. And so that is an H in brackets for a reduction. And so now your aldehyde is going to be a CH2OH. Now note that the bottom group is already a CH2OH. And so now you have a possibility of being symmetrical. In this case, it is not symmetrical because this was from D-glucose. How do you know it was D-glucose? Because of where the OHs are. This is where the OHs are on your D-glucose. So we reduced our D-glucose, and we took our aldehyde and converted that, we reduced it to a primary alcohol. All right, and so this is not symmetrical, but you could have some that would be completely symmetrical now if all the OHs were on the same side, and now the top group and the bottom group are the same. You could have a mirror image. All right, and so your carbonyl is converted to an alcohol, and so it is called glucitol, which means you made your aldehyde into an alcohol. All right, and so these are in gum. You have your D-glucitol, also called sorbitol. All right, so D-mannose. Again, you would not have to memorize D-mannose, but you have memorized your D-glucose. D-glucose has the OH here, 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 and here. It's not here anymore. So this is the C2 epimer of D-glucose. So if I want D-mannose, I will say it's the C2 epimer of D-glucose. That means that the second carbon, we have switched the H and the OH. Again, if we oxidize it, if we do an oxidation, we have our carboxylic acid. If we reduce it, we convert that to a primary alcohol. All right, everything else remains the same. So we don't change anything down here. When you oxidize it, that becomes a carboxylic acid. Everything else is the same. When you reduce it, this becomes a primary alcohol. Everything else down there remains the same. Again, you oxidize it, you get your carboxylic acid. Everything else the same. You reduce it, primary alcohol, everything else exactly the same. All right, some disaccharides, sucrose being the most common. If somebody wants some sugar, they don't want one of hundreds of sugars, they want some sucrose. Sucrose is a disaccharide made of glucose and fructose. All right, so here are some important disaccharides. Okay, they are ones that consist of two monosaccharides. All right, so if you have a maltose, you have two glucose units. If you have lactose, you have a glucose with a galactose. And when you have sucrose, you have glucose and fructose. So here you are making the disaccharide, all right, by a condensation reaction. You simply lasso water from two glucose units, you get a maltose. You lasso water from a glucose and a galactose, you get a lactose. You lasso water from a glucose and fructose, you got a sucrose. Or, of course, you can do the reverse reaction and do a hydrolysis, which means you're going to split with water. You split your maltose with water, you get glucose and glucose. You split lactose with water, you get glucose and galactose. You split sucrose with water, you get a glucose and a fructose. All right, so maltose, malt sugar. You got your two glucose molecules. All right, so now we're going to talk about the alpha glycosidic bond. So it is linked by an alpha 1,4 glycosidic bond. All right, so you have an alpha OH and you have your C4 on the second glucose, and we will hopefully show that in the next one. So this is used in cereals, candies, and brewing, found in both the alpha and the beta form for your, so the alpha OH on one of them, the C4 on the second glucose, and that could be an alpha or a beta form on the second one. So hopefully they show it. Here we go. We got our alpha D-glucose, alpha D-glucose. So we have another alpha D-glucose, two of exactly the same. It's alpha, so I circled the OH to make that one alpha. All right, and so the others to be glucose on C2, you got down, you got up, you got down, you got CH2OH up. All right, over here we have another alpha D-glucose. OH down, that makes it alpha, and then it's glucose because you got C2 down, up, down, and up. All right, so when it says that it is a 1,4 glycosidic bond, that tells you where you're lassoing the water. So the water is lassoed between the C1 and the C4 of the other glucose unit. And so we are going to lasso water here. All right, so the water is gone, so they didn't write that you also get water. All right, so you're lassoing water between your C1 and your C4. All right, and so since this is your anomeric carbon, all right, and that is going down, so that's why it's called an alpha 1,4. 1 carbon number on this side, 4, 1, 2, 3, 4 on this side. So it is a 1,4 glycosidic bond, but it is a alpha because it is going down here to that O. So this carbon that's attached to O is going down. All right, and it said it could be in the alpha or the beta form. And so that means that on this side, all right, so that could be alpha here, or that could be beta. But the glycosidic bond has to be an alpha 1,4. The other OH here could be alpha or beta. In this case, it is the alpha maltose. If the OH was going up, then you would have beta maltose. Both of them have to have an alpha 1,4 glycosidic bond. All right, so lactose, disaccharide of beta D-glucose and alpha or beta D-glucose. All right, so you have to have one that's beta. The other one, it doesn't matter. So you got one that's beta, and then the other one here, they're showing it as alpha, but it could be beta as well. So see, the one has to be a beta, lactose, beta D-galactose. For the glucose, it can be alpha or beta. All right, so beta D-galactose. How is that different than glucose? It is a C4 epimer. So it is down, it is up, it is not down this time. It has been switched to up. We are talking about the C4 epimer of D-glucose, which is D-galactose. All right, but your glucose unit can be alpha or it can be beta. All right, so it doesn't matter about the glucose. The galactose has to be beta, which means your anomeric carbon, the OH, has to be up. All right, and so we're going to have a beta 1,4 glycosidic bond. So your beta 1,4. So now you have your OH and your H. Again, you're going to lasso water, but now this has to be slanted up for a beta glycosidic bond. All right, so that is a beta. And this one, in this example, is an alpha lactose, which is a disaccharide. You could have started with the OH up, and then the OH would be up, and you would have a beta lactose. So you can have either alpha or beta lactose. But your galactose has to be the beta D-galactose in your lactose. All right, sucrose. So sucrose is from an alpha D-glucose and a beta D-fructose. So no other choices. Alpha D-glucose, beta D-fructose. All right, and so when we look at our alpha D-glucose, so here's our glucose. Alpha means the OH was right here going down. All right, and then we had to have our beta D-fructose. So here's our fructose. Beta means that the OH was going up. All right, and so then we lasso the water. When we lasso the water, that's when we get our oxygen between our alpha D-glucose and our beta D-fructose. All right, and so that is a sucrose unit. That is table sugar. All right, so the two monosaccharides that form when sucrose is hydrolyzed, that is simply your fructose and glucose. And they probably want you to write it in the cyclic form, and they do. Okay, so it's not just a glucose, it is an alpha D-glucose. And it's not just a fructose, it is a beta D-fructose. But their answers are simply it's D-glucose and it's D-fructose. More specifically, it is the alpha form of the D-glucose and the beta form of the D-fructose. All right, you got your artificial sweeteners. So you got your sucralose, you got your aspartame, and you got your saccharin. So let's see, what colors are we? The pink packages are the saccharin, which they say is going to give you cancer, and so they still sell it though. So you have to have a lot of it to give yourself cancer. Aspartame, that's your blue packages, and sucralose, that's your yellow packages. All right, so they differ in sweetness. So you can compare your NutraSweet, your aspartame. 60,000 is the sweetness based on a sucrose reference. So it is 600 times more sweet than your sucrose. So you need very small amounts of your artificial sweetener to sweeten your beverage. But anyway, you can make those in the lab. I support my lab making sweeteners and I use those. All right. So your lactose. So you have to know how to make your lactose. You have to know how to make your maltose. You have to know how to make your sucrose. We just did that one. All right. So you got your glucose and your fructose. Your lactose. If you are lactose intolerant, that's because it is your galactose problem. All right. And then also you have a glucose, and then your maltose is going to be two of those. So your lactose has a glucose and a galactose. Your maltose is two glucose units, and sucrose is glucose and fructose. All right. Should be about it for our sugars. One more. Okay, the polysaccharides. All right. So the polysaccharides we haven't covered yet. So we have all right, so you got your polysaccharides, polymers of our D-glucose. And so here we have our alpha OH down, alpha D-glucose. So amylose and amylpectin starches are made of your alpha D-glucose. All right. Glycogen, which is made of alpha D-glucose. All right, and so we have the enzyme to break down the alpha D-glucose. We do not have the enzyme to break down beta D-glucose. So your cellulose, plants and wood, made of your beta D-glucose. All right, so if you look at your structure here of your glucose, all right, so again, you know it's glucose. You got down, you got up, you got down. All right, so then you got up. So you know you're looking at a glucose unit. It's called an alpha 1,4 because your anomeric carbon is going down here. All right, and it's off of carbon number 4 of the other one. That's your anomeric carbon. And so this is going down, which makes it alpha. It is connected between carbon 1 and carbon 4. And so that's why it is an alpha 1,4, another alpha 1,4. Here is an alpha 1,6. So again, the OH is going down here. So that makes it alpha, but it is connected to carbon number 6. And so you can have an alpha 1,6 glycosidic bond as well. All right, so again, alpha because your anomeric carbon is going down, 1,4 because the anomeric carbon is number 1, and then it's connected to the other glucose at carbon number 4. So that's an alpha, and it is a 1,4 glycosidic bond. All right, there's your 1,4 and your 1,6. All right, so glycogen, polysaccharide, stores alpha-D glucose in muscles. All right, similar to amylpectin, but more highly branched. If you look at cellulose, now we're talking not about alpha, but a beta 1,4 glycosidic bond. And it cannot be digested by humans because humans cannot break down the beta 1,4 glycosidic bonds. They do not have the enzyme for that. All right, and so it is a beta because your OH is going up. It is 1,4 because the anomeric carbon is carbon number 1, and it is bonded to the carbon number 4 of the other glucose unit. And so that is why it's called a beta 1,4 glycosidic bond. All right, so you got your cellulose. You will have to know your alpha and beta, and you'll have to know why it's a 1,4 and a 1,6, but you're not going to have to know which starch has which bonds. All right, so that's our monosaccharides and polysaccharides. Go to 15. All right. All right, lipids. All right, so lipids and fatty acids. All right, so lipids are biomolecules that contain fatty acids or steroid nucleus, soluble in organic solvents, but not soluble in water. All right, so these lipids are non-polar, only soluble in organic solvents, not soluble in water. All right, lipose, where the lipid comes from, means fat. All right, and extracted from cells using organic solvents. Again, it's only going to dissolve in a non-polar solvent. All right, types of lipids containing fatty acids are going to be our waxes, fats, and oils, glycerophospholipids, and prostaglandins. The type of lipids that do not contain a fatty acid is going to be a steroid, which is going to have a 4-ring nucleus. All right, so here is our classes of lipids. We have lipid, fatty acid. So a fatty acid is simply a carboxylic acid with 12, usually 12 to 18 carbons. Could be more than that. It's an even number of carbons, usually at least 12. All right, so a long chain of carbons with a carboxylic acid. That is a fatty acid. All right, so when you have your waxes, you have a fatty acid, which is a long chain carboxylic acid, and then you lasso water with a long chain alcohol. And so that is going to be an ester with a long chain of carbons on each side of your ester, and that is a wax. Glycerol, which is a CH2OH, CHOH, and CH2OH. With a fatty acid. So now you've got 3 alcohols to lasso water with 3 fatty acids, and so you have a triglycerol that has an ester, an ester, an ester. It is a triester. So it is doing the esterification reaction 3 times. So your glycerol here, if the bottom one is connected to a phosphate and an amino alcohol, now you have a glycerol phospholipid. All right, and so in biology, normally you have your polar head, that is your polar head, and then you have your non-polar tail. The tails are the carbons of your fatty acid. All right, and then down here is a steroid. So when you have a 6-membered ring, a 6-membered ring, a 6-membered ring, and a 5-membered ring, you have a steroid. That is called the steroid nucleus. If you have that 4-ring system, 3 6-membered rings and 1 5-membered ring, now you have a steroid, which would include cholesterol, bile salts, and steroid hormones. All right, yeah, so 12 to 18 is what we say for a typical fatty acid. That's how many, and it's going to be an even number of carbons. So 15 is not a typical fatty acid. It is always an even number of carbons. 12, 14, 16, or 18 carbon atoms are the typical fatty acids. All right, and so you have your different types of fatty acids. You will have saturated, which has no carbon-carbon double bonds, or unsaturated, which have carbon-carbon double bonds. And of course, you do not want the carbon-carbon double bonds to give you this conformation. All right, so you have your trans fats. Trans fats are bad. You do not want trans fats. So trans fats means that your carbon-carbon double bond are in a trans configuration. What you want and what vegetable oils have is carbon-carbon double bonds that have cis double bonds. So this is what we're looking for. We want cis carbon-carbon double bonds. We want unsaturated fats. All right, so you have saturated, everything's a single bond. You have unsaturated with 1 or more double bond, and you want those to be in a cis configuration. All right, so when you're saturated, you're going to be a solid. All right, when you are unsaturated, you're going to be a liquid. If you're a solid, you're a fat. If you're a liquid, you're an oil. All right, and so that is the difference. And note that they are showing that these carbon-carbon double bonds are going to be cis, because that will put kinks in the chain. You can't pack them, and that's why they are liquids. All right, we will pick up there tomorrow.